G proteins, also known as guanine nucleotide binding proteins, are a family of proteins that act as molecular switches inside cells. The G protein system is the most common method of signaling within cells. Thousands of G protein coupled receptors have been found in our cells, each waiting for its own particular messenger. Their main goal is to transmit signals from a variety of stimuli outside the cell to its interior, as many ligands are polar and able to travel through the semi-permeable membrane of the cell. It is estimated that about 30% of the modern drug cellular targets are GPCRs. The human genome encodes roughly 800 G protein coupled receptors, which can range from detecting photons of light, hormones, growth factors, drugs, and other endogenous ligands, including epinephrine. Approximately 150 of the GPCRs found in the human genome have still unknown functions. All G protein coupled receptors contain 7 membrane-spanning regions, with one end on the outside of the cell and the other inside. G proteins bind to the inner surface of the cell membrane, holding them close to their receptors. Several small lipids are attached to the protein chains, which insert into the membrane and keep the protein attached to the membrane. G protein are made up of alpha, beta, and gamma subunits. In addition, the beta and gamma subunits can form a stable dimeric complex known as the beta-gamma complex. To produce the fight or flight response, the hypothalamus activates two systems, the sympathetic nervous system and the adrenal cortical system. The sympathetic nervous system uses nerve pathways to initiate reactions in the body, and the adrenal cortical system uses the bloodstream. The combined effect of these two systems are the fight or flight response. The sympathetic nervous system sends out impulses to glands and smooth muscles and tells the adrenal medulla to release epinephrine and norepinephrine into the bloodstream. Epinephrine serves as an efficient messenger that signals many cell types throughout the body with many effects. In the lungs, epinephrine binds the receptors on smooth muscle cells wrapped around the bronchioles. This causes the muscles to relax, dilating the bronchioles and allowing more oxygen into the blood. When a ligand binds to the stimulatory G protein coupled receptor found in the muscle cell, it induces a conformational change in the receptor that allows the receptor to function as a guanine nucleotide exchange factor that exchanges GDP for GTP. When GDP is bound to the alpha subunit, it is considered inactive. Thus, when the GTP binds to the alpha subunit, it turns the GPCR on. In this case, epinephrine binds to the G protein coupled receptors. The exchange between GDP and GTP causes a dissociation of the G alpha subunit, which is bound to GTP from the beta gamma dimer and the receptor as a whole. After separating from the beta and gamma dimer, the alpha sub with its bounded GTP then moves along the cell membrane until it finds the enzyme adenylyl cyclase. When the alpha subunit binds with the adenylyl cyclase, it activates the enzyme, causing it to convert ATP into cyclic AMP and pyrophosphate. This happens when ATP drops 2 of its phosphates. cAMP is a secondary messenger used for intracellular signal transduction, such as transferring into cells the effects of hormones, in this case epinephrine, which cannot pass through the plasma membrane. cAMP is also involved in the activation of protein kinases. The main purpose of protein kinase A is to phosphorylate other proteins, which usually lead to activation of another protein. This represents a method in which cells can turn on chemical reactions rapidly and controllably. In the absence of cAMP, protein kinase A is present in the cell in an inactive tetrameric form consisting of 2 catalytic or C subunits and 2 regulatory or R subunits. The R subunits usually block the active sites of the C subunits. Thus, cAMP is needed to activate the protein, allowing cAMP to act as a gatekeeper to control the activity of the protein. Once 4 cAMP are converted using the adenylyl cyclase and binds to the regulatory subunits of a protein, it releases the catalytic portion. The catalytic portion will then add a phosphate to the phosphorylase kinase by using a phosphate from ATP, turning one ATP into ADP. By attaching an additional phosphate onto the phosphorylase kinase, it activates the protein, as phosphorylase kinase is regulated allosterically and by reversibly phosphorylation. The main purpose of phosphorylase kinase is to add phosphate group to the glycogen phosphorylase. Similar to protein kinase A, it will use 1 ATP to add a phosphate on the glycogen phosphorylase, transforming it from a less active state, glycogen phosphorylase B, into a more active state, glycogen phosphorylase A, as well as stabilizing its active conformation. Glycogen phosphorylase's main function is to catalyze the hydrolysis of glycogen to generate glucose 1-phosphate and shortened glycogen molecules. While this glucose 1-phosphate cannot be transferred outside the cell due to its high polarity, it does, however, allow it to be used for intracellular catabolism. With more glycogen phosphorylase active, it enables large quantities of glycogen to be broken down into glucose 1-phosphate, which can then be used for immediate use for the muscle cells. Thus, it is a fast way for muscles to gain the necessary energy during a fight or flight response. To trigger a response in a cell, a large amount of cAMP must be produced in order to change the function of the cell. Thus, the hormone signal must be amplified in order to generate a sufficient second messenger from the few thousand beta-adrenergic receptors present in a cell. A single receptor hormone complex can cause around 100 inactive G protein molecules into their active form. Each active G protein, on the other hand, can only activate a single adenylyl cyclase molecule, which then catalyzes synthesis of many cAMP molecules during the time the ligand is bound to the receptors. Although the exact extent of this amplification is difficult to measure, binding of a single hormone molecule to the G protein molecule can result in the synthesis of at least several hundred cAMP molecules before it is deactivated. Successful cell-to-cell signaling also requires that the response of target cells to a hormone terminate rapidly once the concentration of the hormones such as epinephrine decrease. Termination of the response to hormones with the beta-adrenergic receptors is facilitated by a decrease in the ligand, in this case epinephrine. Thus, less G proteins will be able to convert from an inactive to active form. Within a G protein, the GTP that is attached to the alpha subunit will eventually hydrolyze, exchanging GTP into GDP, allowing the alpha subunit to reassociate with beta and gamma subunits, allowing for a new cycle to occur. However, by itself, the speed at which the GTP is hydrolyzed is too slow for the efficient cycling of G proteins. Therefore, a group of proteins called regulator of G protein signaling act as GTPase accelerating proteins, which promotes GTP hydrolysis, turning GTP into GDP, thus deactivating the G protein, allowing the cell to either activate again or stay inactive. Mutations in G proteins are involved in several different diseases. They can range from whooping cough and cholera to several endocrine disorders. Protein-related diseases are characterized by either deficient or excessive G protein signal transmission, which arises through abnormal signal initiation, defective termination, or reduced levels of G proteins. One disease is cholera. The cholera toxin is a hexameric protein produced by a bacterium known as Vibrio cholerae. The symptoms of cholera include massive diarrhea due to the water flow from the blood through intestinal epithelial cells into the lumen of the intestine, causing dehydration which may lead to death. The alpha subunit of the G protein is irreversibly modified by an addition of an ADP ribosyl group. The modified alpha subunit will still be able to bind with the GTP, but will not be able to hydrolyze it into GDP. Thus, it cannot return to an inactive state. As a result, there is an excessive and non-regulated rise in the amount of cAMP within the cell by 100-folds or more. The rise in cAMP causes the epithelial cell within the intestine to allow massive flow of water from the blood into the intestinal lumen, causing fatal diarrhea.